Hi, I'm Casper Mossman. I'm a communications director at QB3. And today it's a great pleasure to welcome Vasant Jadhav, Senior VP of Research at Alnylam Pharmaceuticals, based in Cambridge, Massachusetts. So a little background, Alnylam was a spin-off apparently from the Max Planck Institute for Biophysical Chemistry. It was launched in 2002, co-founded by about 4 scientists, I think. And it develops and markets RNA interference therapies for a range of conditions. It seems mostly at the moment that's genetic medicines. It has 3 FDA-approved products in the US. By the way, correct me at any point if I'm wrong. 2 other commercial products elsewhere in the world, and a pipeline of 11 compounds in clinical trials at various stages. Dr. Jadhav has over 20 years experience in chemical biology research of nucleic acids, spanning academic, biotech, and big pharma industry for basic diagnostic and therapeutic applications, and a proven record of delivering insightful breakthroughs as an individual contributor, although he doesn't work in the lab anymore, now growing as a team leader overseeing several large multidisciplinary teams. Dr. Jadhav optimized siRNA technology and conceived novel designs as key assets of Rna Therapeutics. And he's been essentially with the same science all the way through since his grad work at the University of Colorado Boulder. Started out with, okay, let me get this right. Rna was acquired by Merck, and then the assets were sold to Alnylam, and Dr. Jadhav followed those. He initiated, established, and oversaw biology efforts of Merck's industry-leading Rna siRNA conjugate delivery platform, and led comprehensive efforts to unearth novel ways of enhancing cytosolic release, which is the biggest barrier to fully realize the potential of the technology. And he earned his PhD from the National Chemical Laboratory in Pune, India, and completed a postdoc at UC Boulder. Dr. Jadhav, it's great to have you.
Thank you, Casper. Let me start sharing the screen here. So thank you for that kind introduction. So for today, I will go through the efforts of Alnylam and in general, the field of RNAi therapeutics. So the title of my talk is Delivering on the Promise of RNAi Therapeutics. So the 3 areas, for the first one is more of an introduction, the RNAi, the Alnylam as a company and the technology. Then the key one being the building the pipeline through delivery advances, and now expanding the pipeline with the extrahepatic delivery. So the in vitro data that really started in Alnylam was in 2002. As you guys recall, in 1998 was the discovery of RNAi by Fire and Mello, showing that the long double-stranded RNA can do RNA interference in a sequence-specific manner. But to really put this in the mammalian cells and do it with the short interfering RNA was really done in this work in 2001 by Tom Tuchel and collaborators, and that really started Alnylam in 2002. So Tom, along with Phil Sharp, Phil Zamore, David Bartel, these were the co-founders who started the company. Now, we are in Cambridge and now global. We have many offices around the world now that we're a commercial company. But the key point here is we took this Nobel Prize-winning science starting in 2002, and 16 years later came with our first approval of the RNAi-based therapeutics. So what took so long? What happened in between? That's the story I will share with you in next 35 or 40 minutes or so. So before going into that, and I won't go through many of these other features, but to say the RNAi is a natural mechanism, evolutionary conserved mechanism that uses this pathway to modulate gene expression. The microRNAs, the natural counterparts of the sRNAs, their function is to modulate the gene expression. So we're essentially doing the same thing. We are using the nature's pathway with exogenous sRNA to control the gene expression of the disease-causing transcripts. Now, this was the promise early on. There was a lot of enthusiasm, but it took a long time to figure this out. And why it took so long time? Well, the key thing was in the field of RNAi, and this was pretty much anticipated in the beginning as well, that the challenge is going to be the delivery. And the reason for that the siRNAs are large. They are highly negatively charged, they are hydrophilic, they are unstable in biological matrices, their nucleus unstable, and many of you working on the RNAs know that how unmodified RNA is so unstable. On top of that, they are immunostimulatory. So they don't really have any drug-like properties. So how do you take the molecule like this and make a medicine out of it? And over the years, and this is really the story where in the early part of Alnylam's and the field's work, the delivery was the key focus. And so we've come up with these two main approaches. The first one being the lipid nanoparticles, where it's a multi-component system where the siRNA is encapsulated and it goes largely in liver. And with this intravenous dosing of these kind of lipid nanoparticles, we have our first ever approved drug, and I'll talk about that a little bit later on. Just another thing to add here, we are the first ones to show that lipid nanoparticles can be used for the delivery of nucleic acids and do the development all the way to get the approved drug. And this is one of the quotes from the magazines, which says that without these lipid shells, there would be no mRNA vaccines for COVID-19. So we feel like in an indirect way, we helped out with these efforts. But the other area where we have put, or the delivery approach where we put most of our attention, is the GalNAc siRNA conjugates. And these are single chemical entity. So this is nothing but the siRNA with the targeting ligand. We'll go a little more details of that one. We have taken this technology and evolved it over the time to now have an Icaria platform. I'll talk about that in the subsequent slides as well. So really, with the promise and solving the delivery is what brought us to where we are today. So building the pipeline based on this one. Let's begin with the lipid nanoparticles and the evolution of these lipid nanoparticles. So the first generation lipid nanoparticles were based on Delian DMA. And with these molecules, we were able to get the human POC to show that RNAi works in humans, but it wasn't really ready. This kind of system wasn't really ready for taking it all the way deeper in the clinical development. Then we got this second generation LNPs. They had much improved potency, almost about 100-fold improvement compared to the first generation ones. These were clinically validated, and now we have the approved drug based on this one. We have continued work with this LNPs and what we call now the next generation ones. These are RE-LNPs, or rapidly eliminating LNPs. So the lipids here are designed in such a way that once they are taken up, they are rapidly degraded inside the cell. So once they have done the job, delivered the siRNA cargo, the lipid is going away pretty quickly. So these are the kind of molecules that we have developed, the system that we have developed for the lipid nanoparticles. And here is one example. And so this is for our program patisiran, and the approved name of this drug is ONPATTRO. This is for polyneuropathy caused by the misfolding of transthyretin or TTR protein. And in this phase 3 Apollo study, we met the primary endpoint, and this was looking at the score, MNIS+7 score, which is the score to understand the polyneuropathy. So as you see in the placebo group, this score kept increasing, and with the treatment group, we saw the improvement, and the p-value was just spectacular here. So we achieved our primary endpoint and all secondary endpoints as well, and this was approved in 2018 as the first ever RNAi therapeutics drug. Now let's move into the GalNAc siRNA conjugates. As I was saying before, with siRNAs, they are incredibly unstable, immunostimulatory. So how do we make these molecules go inside the cell? That's where the site of action is. The RISC, or the RNA-induced silencing complex, the heart of RNAi machinery, is in cytosol largely. So you have to reach all the way, not in any other vesicle or compartment of the cell, but into the cytosol and access this RNA-induced silencing complex. And once that is done, then the siRNA and the RISC knows how to do the job of finding the target and cleaving the target. So with the GalNAc siRNA conjugate, the targeting ligand here is this sugar. And we have this multivalent tri-GalNAc sugar optimized for the recognition by the acyloglycoprotein receptor, which is largely expressed pretty much exclusively on the hepatocytes. In early days, when we're using these molecules, the siRNA stability wasn't there. So we're not really seeing much of an activity. But over the time in the late 2010 or so, in that time frame, we got this, what we call the standard template chemistry, where the siRNA was fully modified. Now, the important thing here is that not only you have to modify the siRNA to improve the metabolic stability, but do it in such a way that it is still recognized by the RNAi pathway. And that was really the key, the secret sauce of achieving that kind of balance of improving the metabolic stability while retaining the recognition by the RNAi pathway. And in the process, we also saw this benefit that these chemical modifications, which are largely done on the sugar part of the 2'-hydroxyl of the siRNA, and a few backbone modifications with the phosphorothioate. So with the standard template chemistry on the left side, we saw the POC that we indeed have the activity in humans with these GalNAc siRNA conjugates. Then by realizing that we can make these things better using further improving the metabolic stability, what we got was the enhanced stabilization conjugate and now the ESC plus conjugates. I will go with some of the examples of the data of these ones, but these are really the basis of the pipeline that we currently have. One of the key things that we found with our standard template chemistry, which is shown here on the left side on the top part, the modifications that we used were the 2'-fluoro and 2'-O-methyl. The GalNAc is on the end, is on the same strand. With these modifications, every residue here is modified. There is essentially no RNA nucleotide here. It's either 2'-fluoro or 2'-O-methyl, and having in a certain pattern that is still recognized by the RNA-induced silencing complex. What we realized with these molecules, although we were seeing the activity, it was quickly getting degraded. It was plasma stable, but not really stable in the intracellular space. So that means it was getting in and getting cleaved, and so the activity was very short-lived, and the potency wasn't very good. And the nucleus hotspot, it appeared that they were coming from the end. So it appeared that this molecule was degraded by exonucleases. So to protect it from exonucleases, we added these additional modifications at the ends of the molecule, and these are shown by these small lines. These are the phosphorothioate modifications that were added towards the ends of the molecule, and by putting these only 6 phosphorothioate linkages, not across the molecule, but at just very few positions, we were really able to improve the exposure, which is shown on the right side here, the difference between the STC and the ESC, the grayish one and the blue one. And with this exposure difference then, it really improved the potency and the durability of our molecules. Now, here is the example. There is a whole lot of preclinical data that has gone through this one. I'm not going through that, but this is directly the human data. So we have 2 molecules here. One is in our early generation or the standard template chemistry, and the other one is in the enhanced stabilization, the advanced ESC chemistry. Same siRNA sequence, same target, obviously, but PD is really different in humans. So with the STC molecule, we were dosing 500 mg every day, 5 days, and after that, a weekly dose 5 times. So nearly 4,500 or 5,000 milligrams of compound. We were seeing knockdown, but this knockdown wasn't staying for a very long time either, as after you stop dosing, it was coming back up. With the advanced ESC version of the same molecule, now the dose is just 50 milligrams, single dose. We achieved same level of knockdown and the durability that is going on for 280 days or so, and so incredible potency and the durability of this with this kind of platform. So we are taken with these platform number of targets, and so far about 10 of them in human clinical studies. And every one of these GalNAc siRNA conjugates have shown target knockdown in the phase 1 studies and beyond, whichever has gone further, most of them have. The only one which is not included here is HBV01, because we did that in the healthy volunteers, so there wasn't really a way to look for the knockdown of the intended target. So across the board for different liver targets for different indications, we have seen very reproducible response in humans, and it's pretty much predicted based on the data that we see in the non-human primates. So we feel very confident when we get the data in non-human primates, how this will translate in humans in terms of the RNAi activity. Now, one of the shining examples of the molecules that we developed with this technology is inclisiran. So the inclisiran is the siRNA targeting PCSK9. This study was done by our partner. We had licensed this out to medicines company, which is now acquired by Novartis. And this drug is now with Novartis. Its trade name is Leqvio. It is approved by EMA and awaiting the decision from FDA this year. The key thing that we found with this molecule was by targeting PCSK9 with inclisiran, first 2 doses given 3 months apart, and after that every 6 months, there was reduction of LDL cholesterol by 54% or so across the board. As it was pretty impressive to see an agent like this one given with subcutaneous dosing, very infrequent dosing, and seeing this kind of response for the LDL cholesterol. And the safety profile was similar to the placebo group. And this was in a pretty big size of trial. About 3,500 or so patients were enrolled in this study. So we're very excited with this kind of data and the profile of the molecules. Now, you might be wondering, well, where is this durability coming from? Is the siRNA showing activity that long? Where is it? What is the reason for this one? So we sort of wonder about different hypotheses here. Is it at the injection site, sub-Q dosing, or is it the siRNA half-life within the RISC itself, the RNA-induced silencing complex, or is it residing within the hepatocytes and slowly coming out of that one, the intracellular depot, which is what we believe is the reason. We believe that there is intracellular depot of the siRNA after it gets into the hepatocytes, and it doesn't really have a way of getting out of that intracellular depot. Imagine the GalNAc siRNA getting into the endocytic pathway and is in endosomes moving into the lysosomes, but then this siRNA doesn't have the way of coming out of these lysosomes. So it is kind of opportunistic, we believe, between fusion and fission events, it might be coming out. But given the potency of these molecules, that you just need very few siRNA copies to get into the RISC and show the activity, we think it is sufficient to sustain the activity. So I will just show you one example of the data that supports this hypothesis. So in the preclinical model, you take GalNAc siRNA and inject by subcutaneous injection. And now you're looking at a picture where we're seeing where is siRNA located, and it co-localizes with lysopenter. So it makes sense, taken up by the GalNAc ASGPR, the receptor-mediated endocytic pathway. So now we wondered, what happens if we tap into these vesicles with an endolytic agent? So we have this peptide that we discovered is an endolytic peptide, small peptide, that was also given using the same targeting ligand, GalNAc, and it was given at different time frames, at 8 hours, at 11 days, or 21 days. So if the siRNA is in that depot, and if this peptide is able to release it, we should be able to see improvement in activity. So let's look at that data. So this is the first one. We're looking at the siRNA conjugate itself. So we're seeing about 70–80% knockdown that is going on for a month or two. Now, you take a second group in which 8 hours later, the peptide was injected. And as you see here, most of the sRNA now we believe is released. It is not trapped in these vesicles, intracellular vesicles. And so it is available in cytosol and it is engaged by RISC, and you see much better knockdown. Well, what if you go later time point? So we went at day 11. Now, by this time point, there is nothing circulating in plasma, and the sRNA plasma PK is very short. It goes into the hepatocytes very, very quickly, in 4 hours or so. But day 11 or so, now we're truly looking at what is inside the cells. And again, as we inject this GalNAc endolytic peptide, we see this boost in activity at day 11. In fact, when we do this at day 21, again, we see that kind of boost. And we have a number of other lines of evidence that suggest that we do have this intracellular depot that might be the most likely the explanation for the duration of activity. So all of this work was published in Nucleic Acid Research. In fact, this article was selected as the paper of the year by Oligonucleotide Therapeutics Society this year. So I encourage you to look into it if you have questions about this one. There's a lot more data about this in that paper. So in addition to the durability, not everything was well. There were some other things that were happening as well. So there were few sequences for which in humans, we saw this transient reversible ALT increases. These were small numbers of the ALT increases, but they were there. So we hypothesized that this might be happening because of noisy sRNA. And what I mean by noisy sRNA is that they might have microRNA-like seed-mediated binding to off targets. So not the full sequence, but the small seed region binding to other targets and doing microRNA-like activity. And that might be the reason for this toxicity in a way. And we saw this one in the preclinical models. But obviously, we select the compounds that have nothing, no these kind of features. So to overcome this, we came up with the mitigation strategy, and that was to use a seed destabilizing modification. And the word seed I'm using here is a region of sRNA that is responsible for the initial binding of the target, the initial binding. So these are first few nucleotides on the 5' end of the sRNA, or the guide strand or the antisense strands, about 2 to 8 or so. So if we can minimize the binding affinity to this region, then it is possible that the off-target effect should go down. But in case of the on-target, which not only recognizes the seed region, but it recognizes the whole sRNA sequence, all 19 nucleotides or 21 nucleotides for the target binding, that if we have this destabilization within seed, that should be tolerated for the on-target. And that's what we saw. We retained the on-target activity, but by having this kind of modification within the seed, this is the glycol nucleic acid, we were able to get rid of the safety findings that were in the preclinical species. And in the RNA-seq experiments, we saw nothing but the on-target knockdown, very little off-target effects. Now, this is preclinical data. Well, ultimately, what is important is what happens in humans. So one of the examples was our program ALN-HBV. So ALN-HBV, when it was tested in healthy volunteers, we saw, as I was saying before, this transient LFT or ALT increases, and it appeared to be kind of dose dependent. And at 3 mg/kg, we're seeing 3 to 7-fold increase over the upper limit of normal for the ALT. Now, we took this compound after doing extensive preclinical work, and this work is published in Nature Communications on all the preclinical work. So we applied this concept of the ESC plus by putting the GNA. So this molecule, the ALN-HBV, the only change we did was to add this seed destabilizing modification. Rest everything else remains same, and we went back. And this program is with Vir Biotechnology, which is in San Francisco, and their data is shown here on the right side. That at 1 to 3 mg/kg, we saw no indication of ALT increase. In fact, at 7 or 10 as well. Only when you go to like 15 mg/kg, you begin to see a little bit of upstick here. Again, that is very small in terms of 1 or 2 fold than the upper limit of normal. So we were very pleased that we took this data from the preclinical experiments, the data from what we saw in clinic, went back with the same compound with a slight modification and essentially fixed it. And so this is the kind of design we call ESC plus, which has improved specificity, so very little or minimal off-target effects, and largely the on-target activity. Now, we've taken all of these advances on siRNA design, on improving their metabolic stability, their potency, their duration, and their specificity, and putting it all together in what we are calling it as Icaria platform. So Icaria is a Greek island where many people live very long life, centurions. So we look at our compounds, we want to see very long lasting activity for depending on the indication, and looking for the designs that will have the potential to have a single dose given every year. So once a year dosing. Go to your doctor, get your shot in a way, and control the disease. So here is one example for the TTR. We've taken TTR sequence on the left side. We're showing the RNA-seq data. It's pretty stunning that there is hardly anything changing other than the TTR. This is in cell culture system, 24 hours. So any secondary effects will take some time to show up. But the primary event of knocking down a specific target that is happening, especially is essentially only for the TTR or the targeted gene. It has potency similar to our previous compound, which is looking really good in the clinical trials as well, but is given at lower doses. So we believe that with this kind of molecule and with its specificity, we can dose it to 3 or 4 mg per kg in humans. And based on our prediction, the modeling algorithm that we have, which has become really good because of the consistency and predictability of the siRNA activity, that we expect more than 90% knockdown of TTR if we give this molecule every year, once a year. And that should have the vutrisiran-like therapeutic benefit. So this is the latest and greatest kind of design that we have on the siRNAs, a molecule which is extremely potent, very, very specific, and can show activity given once a year, simple sub-Q injection. Now, let's kind of review some of the features, just putting them all together. So with RNAi, almost any RNA will be targetable. There isn't anything like undruggable targets that we see with the small molecules. We can achieve very high level of knockdown. And one of the very, very important features is this clamped pharmacodynamics. That means once the siRNA is given, the activity just remains at that level for a very, very long time. If you take small molecules, or in some case of antibodies, there is this sawtooth response because the PK of the small molecules won't be very long. The activity is there as long as the drug is there and it goes back. So there is up and down of the activity, but nothing like that happens with the siRNAs. And it's very infrequent dosing, as low as 2, or now we think as low as 1 dose per year, wide therapeutic index. And now we have this extensive human safety experience across multiple indications, across multiple targets. And this is really leading us now to think about and go into the broader population. But before going into that one, these are the approved drugs from Alnylam. So the first one was ONPATTRO, which is based on lipid nanoparticles. The next 2, the GIVLAARI, which is for acute hepatic porphyria, and OXLUMO, which is for the primary hyperoxaluria type 1. These were approved in 2019, 2020, respectively. And the Leqvio, the cholesterol lowering drug, is approved in EU and is now awaiting the decision from FDA. And this is with Novartis. So with the discovery in 2002, huge amount of work over 16 years or so that led to the first drug, And now with the maturity of this platform, we have more and more drugs coming in our pipeline and looking more and more promising. So where are we going with these ones? So we started with TTR and other compounds like GIVLAARI and OXLUMO. These were more in rare indications. Now we are moving into the specialty indications like cardiomyopathy in ATTR. I mean, these are the trials that are going on now, right now. But then really where we are going now, encouraged by the data that we saw with the PCSK9 or this inclisiran compound, more in the prevalent disease population, where we can take the unique benefits of RNAi technology and can really bring this very meaningful new class of innovative medicines. So I'll give you one example about this one, and that one is on hypertension. We think that with RNAi therapeutics, we could potentially reimagine the treatment of hypertension. I won't go through all the numbers here. Obviously, a huge issue, huge problem. There are many different drugs that are available for this one. But the trouble is that more than 71% of patients have uncontrolled hypertension despite treatment. And this risk is further exacerbated by the variability in the blood pressure control, lack of nighttime dipping, and poor medication adherence. Big problem, compliance. Despite having some good drugs, taking them on the regular basis is really one of the big problems. And we think with siRNAs, that won't be the case because you will be taking it every 6 months or every 1 year, maybe either in doctor's office or whichever kind of places where the medicine will be given. So once it is there, and you won't have to worry about taking the drug every day or with that kind of frequency. So the target that we are going after for this one is angiotensinogen, AGT in short. So we think that by having the liver-specific silencing of AGT and with the prolonged duration of action, we'll have consistent and durable blood pressure response, infrequent dosing that I talked about, and thus a potential for improved adherence. This molecule has gone into phase 1, and this is the data from that. And as you see on the left side, we are seeing beautiful, as we've seen with our other molecules, dose response is very durable. And on the right side, which is obviously the key data, is the dose-dependent reduction in the blood pressure. And so this molecule is now moving into the phase 2. It is initiated in 2021. So really excited about these kind of indications. And there are more examples of like this that we are working on. Sort of given here, this is our pipeline. I talked about the 3 approved drugs, the 4th one with Novartis, Leqvio approved in Europe. And there are multiple of these molecules which are in the late stages, phase 2, phase 3 stages, many in phase 2, and a robust pipeline that is coming behind this one. And RNAi, obviously not limited to liver. By focusing on liver, we were able to optimize our delivery systems. We were able to optimize the siRNA, understand how well RNAi works in humans, but microRNAs and RNAi pathway, pretty much ubiquitous, work all cell types. So why not go after now, take all these learnings that we have and apply to other places? And so this is showing some of the papers that we have published and the progress that we have done with the conjugates. And on the right side, we're showing here the years and the ED50, the 50% knockdown, and this is the rodent data. So we started with 100 mg per kg in early 2006 timeframe. And over the years, really the breakthrough came with the standard template chemistry in 2010 timeframe, and that we have continued to improve on that one to have the molecules which have incredible potency and specificity. So we're now taking the lessons learned from these molecules and applying it for the extrahepatic delivery. And the areas where we're going in the tissues where we are expanding now more are CNS, eye, and lung. There are multiple disease areas here with high unmet medical need. One of the key things that is in our consideration is having the targets that are genetically validated and have the established biomarkers. It helps with conducting the clinical trials. You don't have to worry about the target as much because there is a human genetics behind that one, and really using the learnings that we have from our program, liver program. And to do that, we have these alliances in extrahepatic discovery and development. The first one now a couple of years ago, we have this alliance with Regeneron. This is in the field of CNS and eye diseases. And we are also now working with a company called Peptid Dream, who makes cyclic peptides. We believe that with these kind of technology, we will be able to identify targeting ligands to go after different targets, receptors expressed on various tissues of interest. So let me show you some of the data in, and just a snapshot of the data in these different tissue types. So here is one from the CNS platform. So obviously we're looking for high potency, wide biodistribution in CNS. Now, unlike in liver, where hepatocyte is predominantly the major cell type, there are multiple cell types in CNS. And if you want to really go and achieve activity in all of them, sort of need a ligand that will go and have activity in all of them. Long duration of action, very important. If you're using the intrathecal kind of dosing, you don't want to do this thing very frequently at all. And obviously want to have the favorable risk-benefit profile. Here is the data in CNS. This is in NHP. So this is for amyloid precursor protein. We're looking at the biomarkers, alpha and beta soluble APP. And with a single injection with this APP siRNA, by intrathecal dosing, we're seeing robust and very durable knockdown. We've done huge amount of preclinical work and identified a development candidate, what we call ALN-APP. The initial CTA submission is expected for this one by the year end. So really very exciting opportunity to see how RNAi therapeutics could impact the diseases where in CNS, like Alzheimer's disease. We are going with the indication first with the early onset to see and test out the technology. So really excited about the progress and looking forward how this works out. The second example in the extrahepatic space is in eye. So here is the data in non-human primate eye, very, very small doses of 0.03 mg or 3 micrograms or 30 micrograms of siRNA conjugate given in eye, showing very robust activity. And that this activity is from day 84. Again, the durability feature remains there in CNS as well as in eye. We believe the similar mechanism is at play here as well. That is the intracellular depot likely providing the siRNA to the risk for a long time to retain the activity. We also have shown very nice data in lung. This is by intranasal delivery, and this is for one of the targets that is expressed ubiquitously, SOD1. So we see wide distribution and the reduction of this mRNA in a dose-dependent manner, and again, which it remains for a very long time as well. We also saw the activity for the human ACE2 receptor, obviously very important one as we think about the SARS-CoV-2. But this was just an example that taking this progress from liver to these extrahepatic tissues, we are now really seeing good activity in these tissues, and we want to expand RNAi therapeutics to many more targets by having the approaches like this. So with that one, so the Alnylam's product engine is, we expect about 2 to 4 INDs per year. There are a large number of opportunities, and this is all organic capability and growth that we have. We're not in licensing these compounds. These are the compounds which we are discovering internally and developing them. So these efforts from the beginning and the investment in making the delivery work is now we're utilizing human genetics, UK Biobank kind of efforts, and all the lessons that we have learned to really open up RNAi therapeutics, not just for liver, but for other tissues to treat tremendous unmet medical needs, especially when you think about some of these terrible CNS diseases or eye diseases. I will just end with this one slide here, that building a new class of medicine obviously takes a long time. And just taking an example of monoclonal antibodies, that the discovery was in 1975, 9 years later was the Nobel Prize in 1984, and the first MAP-based drug was approved in 1994. Now, after the first approval, and in 2018, 6 out of top 10 best-selling drugs seem to be on the monoclonal antibody technology. Well, we'll look at the comparison with RNA therapeutics, obviously early days, but the discovery was in 1998. 8 years later was the Nobel Prize for Fire and Mellow in 2006. 12 years later, we got our first approval. So similar kind of time frames from discovery to the first approval. And since the first approval in 2018, we already have 2 more, and the fourth one, which is approved in Europe with with Novartis. So there is a big expansion of RNAi therapeutics. Time will tell where we'll go from here, but we are expanding it with the intent of addressing much of unmet medical need by going after the targets in different tissues. With that, I will end, and happy to take questions.
Great, thank you so much, Vasant. There are 3 questions that people have asked already. I've got a few of my own. I'll ask the first one. It's from Brad Niles. Can you share your thoughts on concepts and the potential of siRNA for cancer?
Yeah, I mean, so I think as we started before, having the focused effort in liver was very, very important. Solve the delivery there, go after genetically validated targets, monogenic diseases, right? So we can take all of these lessons now and expand it for the field like oncology. Now we have to also be mindful that in oncology, it may not be just one target. So with that in mind, but it's very much in our minds as well. But first, we are going into CNS and ocular with those kind of ones. But ultimately, I would say this is more matter of time rather than will it or not, in my mind. I think I can maybe read through this one. So the therapeutic durability seems like a positive attribute, but if a patient has adverse reaction, is there a way to inhibit siRNA activity? Very, very important question. And so this is where, again, having the human genetics for the targets is important. That means there are people who have loss of function for that particular gene, and they can still have a particular, I mean, there are obviously some detrimental things going on. But that setting that aside, we also have, I mean, this is one of the key benefits of sequence-based technology. Casper, we were talking about it earlier, right? That when you have the technology for proteins, which are rather recognizing the structure, here we are recognizing the sequence, the code. And so if it is a sequence-based technology, you can imagine that you can also make an antidote for it. And in fact, we do have a technology, what we call reversier. So reversier is essentially a short synthetic target, which looks like more like a natural target, but it is small, is synthetic, and it's high affinity. And so by providing it with the same pathway as the siRNA is given, meaning with the same GalNAc pathway, we see incredibly potent, robust reversal of siRNA activity within days. And it comes back to baseline. Now, is it happening within hours? No, it takes days, couple of days in preclinical species to come back, but we do have that approach that is available. Okay, I think there's a question by Carl. maybe just read it out. Beyond relatively unspecific nanoparticle targeting and liver-directed targeting via GalNAc, what methodologies do you think will be best? You know, I mean, everything is open. Now the playbook is there. When we think about the variables here, sRNA being one, and the second being the targeting delivery. Now we can focus a lot more on targeting rather than the sRNA piece, because that part is optimally modified, metabolically stable, and we can go after looking for different targeting ligands. And as I was saying before, we have this collaboration with PeptiDream. These are the kind of approaches that we are taking now to find the ligands that can improve the sRNA delivery in different places. And we're pretty open. It could be small molecules, it could be peptides, it could be nanobody, it could be antibody. We'll take whatever works. They might have their pros and cons. Maybe antibody sRNA conjugates might have improved PK, at least circulating plasma PK. But do we really want that? So we want to have these options, figure that out. I don't know if love the potential for hypertension. Absolutely. I mean, imagine your drug that is coming for reducing the LDL, reducing blood pressure, and the way it does, this tonic way of controlling for a long time. What percentage of liver cells are transfected with one dose? We think most of them, because, and this is based on the knockdown that we get. I mean, getting 90% on higher knockdown for the target. So we think most of them are getting taking the sRNA, and then GPR is abundantly expressed. So, are you interested in sRNA in cancer by in licensing? You know, this is something I would have my BD folks, but these are the kind of things that are obviously on our minds as well. Could you share your thoughts on the Ionis? I would let them comment about this one. We think our technology is uniquely suited, and with its potency and durability, we are very hopeful that we'll be able to address many of the CNS indications that we are going after. Again, early stages. We haven't taken our molecules in humans, but we are beginning with the ALN-APP, and fingers crossed.
Can I ask a question?
Sure.
Do you, again, I know very little about this, but do you, is there the potential either to up or down regulate a gene product by just you know, targeting the expression of a repressor or a promoter? So you can basically take you know, a condition in which the gene might be like over repressed, or might even like have a single copy or whatever, and you can amp up by using siRNA.
Oh, absolutely. In fact, patisiran, which is one of the programs that is with Sanofi, so we developed it, and Sanofi is now conducting the late trials with that one. So the patisiran is a program for hemophilia, and here the goal is to improve the thrombin generation, or the level of thrombin. What happens is the antithrombin is now reducing the thrombin levels. So rather than finding a ways by RNAi to improve or upregulate thrombin, what we are doing is we are going after antithrombin. So by going after the repressor or the brake, we are now able to have higher level of thrombin, and that ultimately helps. And so certainly, because the biology is so beautifully in that way, that you can go in the pathway and upregulate by being upstream, downstream. The second example of this one, very similar approach, a little bit different, is our approved drug, givosiran. This is for acute intermittent porphyria. So in that one, toxic metabolites are forming, but the enzyme which is defective, we're going a step before that one, so that if we don't have that enzyme, the products of that enzyme are not toxic. They are not reaching to the next step. So we are absolutely using these kind of approaches to really use the RNAi.
Can you say a bit more about how you choose the diseases to go after? Obviously, you want one that it's been genetically defined and you've got the biomarker, but how do you choose what disease and then what molecule to go after in that disease?
Yeah, no, it's a great question. So I would say, first of all, in a way, we are disease agnostics, meaning we are going for the targets which are expressed in liver, which is where the delivery works, right? So that's what we're doing. So when you look at our pipeline and our approved molecules, that are not really in a particular, in one class of indication. They're very different from each other. But the common thing is they are liver expressed. They are biomarkers. They have human genetics behind that one. So yeah, I mean, really, that's the way we're approaching it.
Just because you have a very successful way to target it to a tissue.
Exactly, and we hope that we will now open it up by going in CNS, in eye, and in other tissues, because RNAi is just everywhere. It's really, yeah, resources.
So I see nobody else has any further questions. I've got one last one, but it's a rather naive one. How short is short? When you make these RNA strands, like how many of them are there, and what's optimal? How many residues?
So the designs, I mean, we're learning from nature, right? So microRNAs, the natural counterparts of the sRNAs are in the range of 1921 nucleotides or base pairs long. So that's essentially the range of the sRNAs. So 19 to 21 base pairs.
Okay, great. I imagine if they got too long, then they might start getting too complicated to maybe unfold and find their targets.
You know, it might still work, because at the end of the day, it is still a sequence, right? And the initial discovery, obviously, which was by Fire and Mello in 1998, was with a long double-stranded RNA, much longer than these 1920, right? So it was hundreds of base pairs long, and but then once it is there inside the cells, it gets processed by dicer, and you're making the smaller ones. So the longer ones also work, but you don't really need them to be long. You pretty much have the perfect size of that 1921 nucleotides. If you make them shorter than that, then you start losing the activity.
Got it. Okay, one final question for me. Are you hiring? Are you looking for any positions on your team?
Oh, yes. Oh my goodness, please. I mean, I didn't want to make that kind of a pitch there, but yes, we are hiring. We are a company now which has grown from initially, when I joined 8 years ago at Alnylam, we're like 150 people. We're about 1,500 now. We're a company with approved products now, so there is a lot of, obviously, folks all over it's a global company in Japan, Europe, and all different places. But we are hiring, we are growing in a meaningful way. You don't want to become too big, too fast, but we're definitely hiring. I would encourage folks who are interested to please go on our website. All the information is there, but we're certainly hiring.
Great. Thank you so much for speaking to our audience today, Vasant. Really appreciate it.
Okay, I had fun. Thank you very much for the invite.
Bye, everybody.